My main focus of research is adaptive radiation, which is when a species rapidly diversifies into multiple species utilizing different ecological niches. One of the most famous examples of adaptive radiation are the finches on the Galapagos Islands, which have evolved into many different species utilizing different food resources like different seed sizes. So adaptive radiation requires ecological opportunity like the different seed sizes so that the species can adapt to different ecological niches. And it also requires reproductive isolation, some way of allowing the species to coexist together without merging back into a single species. And both of these require genetic diversity. And the genetic diversity is another major focus of my research, as you will see in the following talk. I'm not studying Darwin's finches, though. I'm studying cichlid fishes and butterflies, as Julie also said. But on this talk will be mostly cichlid fish. So you may know these iconic examples of adaptive radiation from Lake Victoria, Tanganyika, and Malawi, where cichlid fishes have evolved amazing adaptive radiations in each of these lakes. However, cichlids don't just colonize a lake and then radiate. Actually, in most cases, when cichlids colonize a lake, they don't form any adaptive radiation. Katie Wagner, a former postdoc of Ole Seehausen, with whom I worked together during my PhD and first postdoc, she studied what predicts if cichlids colonize a lake, if they radiate or not. She found that in about 25% of the cases where cichlids colonized the lake, they radiated. But she also found 145 colonizations where radiations did not happen. So she looked at what factors predict if adaptive radiations happen or not. What she found are major predictors of ecological opportunity, like lake depth and energy, and other predictors of sexual selection, which are important for allowing them to evolve assortative mating and therefore to stay reproductively isolated by choosing to mate with individuals from their own species and avoiding individuals from other species. Recently, we revisited this topic in a study led by Matt McKee, who was also a postdoc in Ole Seehausen's lab and is now an assistant professor at Monash University. He managed to make a phylogenetic tree of all 1,712 described species and used this approach to then study what factors affect speciation rates. And again, he found very similar factors, like a large depth gradient is a positive predictor of high speciation rate, or male ornaments, which relate to assortative mating. Whereas low rainfall and predator presence, two things that reduce ecological opportunity, make speciation rates lower or are correlated with slower speciation rates. So we know already quite a lot about what factors are important also for cichlid fish in adaptive radiations, but there are still some adaptive radiations that are just completely crazy and cannot be explained by the combination of these factors. And one of them is the radiation in Lake Victoria. Lake Victoria is a lake in East Africa. It's a very big lake, and it was colonized by 5 cichlid lineages, these ones here. And these 4 did not speciate at all when they colonized the lake, whereas the 5th one diversified into 500 species. They all arrived at the same time at the beginning of the lake. We know this from sediment core teeth findings. And so it's not that this one used up all ecological opportunity, basically occupied each ecological niche, and then the others arrived later. It's rather something that seems to be specific to this lineage. And this lineage hasn't just radiated in Lake Victoria, it has generated 500 species there, but it has also radiated in Lake Albert, Edward, Kivu, in the Kagera lakes, etc. So in all lakes of this region, this same lineage has radiated over and over and over again. Together, it evolved 700 species and this happened in just 150,000 years, so extremely rapid. This lineage is called Lake Victoria region superflock, superflock because it's basically like an assemblage of flocks, an assemblage of different adaptive radiations. And in each of the lakes, these cichlids occupy all kinds of niches you can imagine, from top predators feeding on other cichlids down to cichlids that are specialized algae scrapers. So in my PhD, I wanted to find out where did these cichlids come from? Who are their closest relatives? For that, we performed rat sequencing of over 100 cichlid species from all across Africa, from major tributaries of the region and all rivers and lakes that could have been in contact with the Lake Victoria region presently or in the past. The phylogenetic tree I generated from this data set is too big to see anything, so let's just zoom into this region here. What I found is that the Lake Victoria region radiation, so this Lake Victoria region superflock, they form a monophyletic group. And the closest relatives are cichlids from the Congo, and the next closest relatives are cichlids from, so the Congo here, from the Upper Nile region and the region on the east of the Lake Victoria region. But when I tested if this phylogeny is a good representation of the ancestry of the Lake Victoria region superflock, I found that this entire superflock here has parts of the ancestral variation derived from the Upper Nile lineage. So when I test for hybridization, I find a very strong signal with both of these Upper Nile representatives. With lots of different tests of hybridization, I identified this scenario where the Upper Nile lineage and the Congolese lineage are both ancestral to the Lake Victoria region superflock and have hybridized about 150,000 years ago, whereby about 20% of the ancestry of the Lake Victoria region superflock is derived from the Upper Nile lineage and about 80% from the Congolese lineage. So Upper Nile and Congolese came together, hybridized, and from this, the 700 species of the Lake Victoria region superflock evolved. But did this hybridization actually facilitate the rapid diversification of this Lake Victoria region superflock, or did it just happen and just have no effect whatsoever? So for that, let's go one step back. Where does genetic variation come from? I said for adaptive radiation, genetic variation is required, both for adaptation to different niches and also for reproductive isolation. So genetic variation is very important in adaptive radiations. It can arise either via new mutations, but new mutations, most of them are deleterious. Most of them have no effect or only very, very small effects. And it is very rare that mutations happen that actually facilitate, for instance, adaptation to different niches. And then comes another problem. Once species has speciated into two species adapted to different niches, the genes that were under strong selection to adapt to these niches are then depleted of genetic variation, and new speciation events in rapid succession become harder and harder. It gets easier if there is already standing variation, where you already have multiple different gene variants in the same population. And then speciation can happen more easily. But again, we have the problem that now the new arising species have again lower standing genetic variation and speciation gets harder again. However, maybe hybridization could help here, because during hybridization, two very divergent genomes or genetic diversities come together, and lots of different combinations of this genetic variation can arise from that. And then different species may arise via sorting of these different gene variants into different combinations, which then form new species adapted to different niches. So if that was the case, what we would expect is that a Congolese and an allotic ancestor, when they hybridize, they formed a hybrid swarm with lots of different hybrids having some parts of the genome from the Congo ancestor and other parts from the Upper Nile ancestry. And then new species may arise by sorting of this ancestral diversity, where each species is a different mosaic of the Congolese and the allotic ancestor. And this is exactly what I found. So I found that at sites that are under divergent selection, some species, like for instance at this position, have fixed the Congolese allele, and other species have fixed the Upper Nile allele. And then when you look across the genome, each species is a different mosaic of the two ancestral lineages. By the way, I have a very weird side noise. Maybe everyone turn off your microphone. For most of these genetic variants, unfortunately, I don't know what they really do, if they facilitate adaptation to different niches, for instance. But for one of them, we do know a lot, and that's the long-wave sensitive opsin gene that allows cichlids to adapt to different water depths, because in the deeper water, the light spectrum is more redshifted, and the red opsin gene is very important, whereas in the shallow water, there's the light spectrum is less redshifted, and a different opsin variant is beneficial in shallow water. And here at this gene, I found that there are two deeply divergent haplotype classes, where different species from Lake Victoria, here shown with orange symbols, some of them have derived the Congolese variant, and others have fixed the Upper Nile gene variant, allowing them to adapt to turbid deep water or to shallow water. So here this shows that bringing together the genetic variation of the Congolese and the Upper Nile lineage facilitated adaptation to different water depths. And you can imagine that this might have happened at many different genes that allow the species to partition the niches in many different ways. And therefore, the combined genetic variation of these two lineages may have facilitated adaptation to different niches. So did hybridization facilitate the rapid diversification? We think yes. The two divergent ancestral lineages provided high genetic variation that was then selected into different species. But this hybridization event was 150,000 years ago, and it probably facilitated the origin of the entire Lake Victoria region superflock, all the cichlids that together occur in this region. But actually Lake Victoria itself is much younger than this 150,000 years ago. And that's because it completely desiccated and refilled only 15,000 years ago. And now it has this amazing diversity of cichlid fish occupying each ecological niche you can imagine. So did these 500 species really evolve in only just 15,000 years since refilling of the lake? To get at that, we did whole genome resequencing of over 450 genomes of all the different radiations in the Lake Victoria region, particularly focusing on the variation within Lake Victoria, but also all major representatives of all major ecological groups from all the other lakes, and to see if the Lake Victoria cichlids maybe have some association with these other radiations. For instance, Lake Edward and Kivu are deep rift lakes which may not have dried out during the dry period. And the Kagera lakes are at high altitude in more humid climate and may therefore still have harbored some water bodies that could have hosted some ecological diversity during the dry period, which may then have colonized the lake as an ecologically diverse radiation. So first, if we look at how the species within Lake Victoria clustered, this is now a PCA of 293 whole genomes. And you can see the different ecological groups shown with different symbols, like here these stars are eggs and fry eaters. here we have a little group of detritivores, or here we have some predators. And it seems in general that the species cluster by ecology. So this could be that the detritivores are derived from one detritivorous lineage that colonized the lake, and the predators are derived from another predatory lineage that colonized the lake. Or alternatively, it's possible that a more generalistic lineage colonized the lake and then made adaptive radiations and evolved detritivores and predators and egg and fry eaters from the same lineage. This is what I wanted to find out. And for that, we now need to compare, for instance, predators from Lake Victoria with predators from the other lakes to see if they are clustering together with predators from the other lakes or with insectivores and detritivores, etc., from Lake Victoria. So when we do that, when we add in the other lakes to the PCA now, we can see that the Lake Victoria and Kyoga cichlids cluster together. So Lake Kyoga is this lake here, which is also extremely shallow and for sure desiccated and seems to be part of one single radiation. These cichlids, they all cluster together tightly. The next closest group are cichlids from the Kagera lakes, which are up here, higher altitude, and might still have hosted some ecological diversity. And they're all distinct from these western lakes, which are these ones here, Albert, Edward, Kiwasaka. And we also see that, for instance, our predators from Lake Victoria clustered together with predators from Lake Kyoga, but are completely distinct from predators of Edward, Kiwu, Albert, or Kagera lakes. So there's no indication that the different ecological groups within Lake Victoria are derived from different colonization events of the same ecological group from another lake. So did the 500 species really evolve in only just 15,000 years since refilling of the lake? Yes, all Lake Victoria species are each other's closest relatives. It seems that they all derived, that they all evolved within the lake. But are really all Lake Victoria species derived from a single colonizing lineage? Or did maybe again multiple lineages colonize and hybridize, as we have seen at the beginning of the Lake Victoria region superflock? This can then afterwards still look like a monophyletic group of genetically very similar cichlids, even if they are derived from multiple lineages, but just through admixing their genomes, they become more similar. When we look at the mitochondrial tree, so this is now the whole mitochondrial genome, we see in orange the Lake Victoria cichlids, in red the Kagera Lake cichlids, and in blue the Western Lake cichlids. And you can see here that the Lake Victoria cichlids form two groups, which are deeply divergent, by far preceding the origin of the lake. So there must have been at least two lineages that colonized the lake. One of them is shared with Kagera Lake cichlids, but they don't seem to be at the basal position. But also there's not very much support, so it's hard to tell. And the other lineage is not shared with anything else, so I don't know where it came from. But what is clear is that there are two deeply divergent lineages. We don't know where they came from, but there were probably two lineages that colonized Lake Victoria. And when we look at the cichlids that have this very divergent mitochondrial haplotype, they fall right within in the nuclear genome. They are not different in the nuclear genome from the other cichlids at all. So this means that the two colonizing lineages, which are still seen in the mitochondrial tree, they must have hybridized, leading to homogeneous genes in the nuclear genome. The most basal clade in the Lake Victoria Kyoga radiation is actually cichlids which are not right within Lake Victoria, but from swamps around the lake. It's this group of Astatotilapia nubila. And I wanted to check if maybe I can find evidence for additional hybridization into the Lake Victoria Kyoga radiation. that would not be shared with the swamp cichlids around the lake. For that, I used the t-statistic, or also called Baba test. One compares sites where Lake Victoria cichlids are more similar to either Lake Edward, Kivu, Albert, or Saka cichlids than swamp cichlids are to Edward, Kivu, Albert, and Saka. So in a positive signal would mean that there was excess allele sharing potential, additional hybridization between Victoria cichlid and any of these, whereas a negative signal would mean the swamp cichlids show excess allele sharing or potentially hybridization with any of these other cichlids. And what I found is that all Western lake cichlids show excess allele sharing with Lake Victoria cichlids. So this means that probably there was additional introgression into Lake Victoria cichlids, but because all of the lakes show the exact same signal, I don't really know which one of them it was. Maybe it was none of these sampled lakes. Maybe it was a close relative of all of them. It's really hard to tell. But there seems to be an additional contribution from Western lake-like cichlids into the Lake Victoria Kyoga radiation. So are they derived from a single colonizing lineage? Not really. There are two deeply divergent mitochondrial lineages, and there's also evidence for additional introgression from the Western lakes. So it seems that multiple lineages combine the genetic variation through hybridization. And that explains why the Lake Victoria Kyoga cichlids are pelagic, despite apparently being derived from multiple colonizing lineages. So we had originally 150,000 years ago a hybrid origin of the entire Lake Victoria region superflock. And then now, about 15,000 years ago, at the beginning of the lake formation of Lake Victoria, it seems that there was another hybrid origin, but now just of the Lake Victoria radiation itself. So less divergent lineages, maybe from different refugial swamp populations, we don't really know, came together in Lake Victoria, hybridized and radiated again. But did the lineages provide genetic variation underlying the ecological groups in Lake Victoria? Like, for instance, did they provide genetic variants which then be selected into predators or algae scrapers? Or did within Lake Victoria new mutations arise and allow adaptation to these different ecological groups? For that, I used an F4 test, which that I used to compare cichlids from the different lakes. So for instance, here I'm comparing cichlids that feed on mollusks and cichlids that feed on other cichlids from Lake Victoria with cichlids that also feed on mollusks or other fish from Lake Kivu. And in most cases, they cluster like this. But in some sites, the mollusk feeders, the snail eaters, they cluster together even though they're from different lakes, and the fish eaters cluster together. And in other regions of the genome, the snail eater from one lake goes together with the fish eater from another lake, etc. And now I'm comparing which of these two patterns is more common. If this pattern is more common, this would suggest that there is some sites in the genome where the snail eaters share genetic variation, which may have been important in making them snail eaters, which are not shared with the fish-eating cichlids from these two lakes. So these would be sites that potentially have been selected into similar ecological groups within Lake Victoria that are old variants which were already important for making the same ecological groups in the older Lake Kivu radiation. And this is indeed what I find. This pattern is more common than that one. And when I did that across many different pairs of trophic groups of different ecological groups, I found in some cases no evidence for excess allele sharing between cichlids that have the same ecological group. But in many cases, I found a very, very slight excess allele sharing. So while in most parts of the genome, the cichlids cluster by lake, there is some allele sharing between cichlids from different radiations that occupy the same ecological niche, suggesting that the same gene variants were involved in making them, allowing them to adapt to the same ecological niche. So did these lineages provide the genetic variation underlying the ecological groups in Lake Victoria? Yes, some genetic variation of the ecological groups in the older radiation seem to have been sorted into similar ecological groups in Lake Victoria. This is what this F4 test showed. But then comes one puzzling thing. So it's not just re-evolution of the same ecological groups, which happened in Lake Victoria. There are also many new ecological groups in Lake Victoria, which are not found in any of the older lake radiations. One example are pelagic dwarf predators, which are little predators that kind of look like a pelagic zooplanktivore, but they feed on fish like the large predators and also have kind of predatory mouth. And this is basically also what they look like genetically. So this is now genetic similarity matrix. When you can see such triangles, this means that all the cichlids in this triangle are genetically very similar. And here we have a little triangle of the dwarf predators, and they form a big triangle with the large predators, showing that they're genetically very similar to the large predators. But there's also some excess allele sharing with the zooplanktivores. So the dwarf predators are genetically partially large predator-like and partially zooplanktivores. So this suggests that even within the Lake Victoria radiation, hybridization between different ecological groups seems to have given rise to new ecological groups via new combinations of the genetic variants in these two groups. At least some of these new groups thus seem to have evolved by combining the genetic variation in novel ways. So this means we can now add another level of hybridization, which is less than 15,000 years ago within the adaptive radiation of Lake Victoria. There is hybrid origin of a new trophic group. So we find that hybridization affected the very beginning of the entire superflock, the beginning of the Lake Victoria radiation after it desiccated, and even within the radiation, new ecological groups emerged via hybridization. So basically, it seems that hybridization and then resorting of this ancestral, of this admixture variation played a very important role throughout the evolutionary history of these cichlids. But now, if hybridization is so beneficial to adaptive radiation, why did these other cichlids not hybridize? Sorry, I have to drink something. And the reason is very simple. These cichlids here, they don't have close relatives. And so hybridization just never occurred. But this lineage here, the Pseudocrenilabrus, it colonized all the different lakes in the region. It never hybridized because there is no closely related lineage nearby. And it never speciated. But it also colonized Lake Mweru and Bangweulu further south. So here's Lake Victoria and here's Bangweulu and Mweru. Further south in the region, a Pseudocrenilabrus representative colonized Lake Bangweulu and didn't speciate either. A Congolese lineage of Pseudocrenilabrus colonized Lake Mweru because Lake Mweru is part of the Congo drainage system. And then about 1 million years ago, there was a river flow reversal, where now cichlids from Lake Bangweulu could swim into Lake Mweru. The other way doesn't work because of waterfalls. But now the Zambezi lineage and the Congolese lineage could come together in Lake Mweru. They hybridized and radiated into 15 species. So it's not that this lineage is per se unable to speciate. I assume in Lake Victoria, there was just not enough time within 15,000 years. Without hybridization, the genetic variation may just not have been big enough for adaptive radiation within just 15,000 years. Whereas in this lake here, where hybridization did happen, within a million years, it evolved into many different species. So basically, you can imagine that the cichlids seem to have evolved here via combining the genetic variants of very different lineages, or the Lego bricks of very different objects, that were then sorted into many different other objects. And this happened then again at the base of Lake Victoria. And via combining two objects within Lake Victoria, a new ecological group like the dwarf predators could evolve. So this is really nice. And I was extremely excited to find hybridization over and over to play a very important role in this radiation. But what I was always a bit annoyed about is that for most of these Lego bricks, or for most genes, we don't really know what they do. And so recently I have switched to working on butterflies. I went to Ecuador just before it wasn't possible to go on fieldwork anymore, and collected Heliconius butterflies, and particularly also ethomine butterflies, which are my new study system. And here it's very convenient because we can see immediately which traits changed because of strong selection on looking the same way like all other local butterflies. And this is because the birds learn to avoid to eat these butterflies. They're toxic. And they learn to associate specific patterns with a disgusting feeling. And so when they see another butterfly, they remember and they won't eat it, if it looks the same as those butterflies that the birds have already tried eating. Whereas if a butterfly looks different, they may try it. And therefore it's a very strong selection on looking like the locally most common butterflies. So in this first project, where I already have results, is a project with Chris Jiggins and Patricia Salazar. Patricia actually collected all the butterflies, and they together started this project. It's here in Ecuador. There is a region between the Amazon and the Andes along an altitudinal gradient, where Heliconius serrato looks like this in the highland and like this in the lowland. And Heliconius melpomene looks very similar in the highland and in the lowland. But for me, most interestingly, in the middle, we get this new subspecies, which combines the red pattern of the lowland species and the four-wing patches of the highland, sorry, not species, subspecies, and the four-wing patches of the highland subspecies in both species. And so I wanted to know if this is another case of hybridization playing an important role. For this, we used haplotagging, a new linked read technology that was developed by Frank Chan and Marek Kuka and Andrea Dreu in his lab, which basically allows you to wrap DNA around beads and then all DNA fragments on the same bead, so that the TN5 transposase, which is on the bead, cuts the DNA and adds a barcode, which is the same one for all DNA fragments on this bead. And then when you sequence the DNA, you know which parts came from the same molecule. And that helps because you can get phased data very easily without having to go for high sequencing depth. And it's also very good for finding structural variants. So we sequenced almost 500 individuals of Erato along this transect, and almost 200 individuals from Melpomene along this transect. When I just compared the highland and the lowland forms of Heliconius erato, I found that there are 4 major peaks. And in Melpomene, I found the exact same 4 major peaks of FSD. So these are regions where the highland and the lowland forms are strongly divergent. And I found the same 4 regions showing up. When I performed a GWAS, so a genetic association study to find which genome regions are associated with specific phenotypes, I found that this high FSD peak here is associated with having 2 or 1 4-wing patch. And this one here, for instance, is associated with having red in the 4-wing patches, or is this lowland-like red pattern. And so all 4 peaks I found to be involved in color differences between the 2 subspecies. And when you zoom into those 4 regions, the parallelism is really surprising, really, really interesting. It's really the same 4 genes that seem to be divergent between highland and lowland forms in Erato and in Melpomene. And when we look at how the gene variants change from highland to lowland, for instance, at optics in Erato, we find that at high altitude, the high altitude variant is fixed. And then as we move along the transect towards lower altitude, there is a steep decline in the highland variant at around 30 kilometers, indicating that there's really like a hybrid zone here. But then when we do the same for Wnt, which is the gene that determines having 2 or 1 4-wing patch, there is a strong shift with a center around 50 kilometers. And we find the exact same shift in both species, in Erato and in Melpomene, which is why we find this new hybrid form in the center, because here in the center, they are fixed for the lowland gene variant for optics, which generates this red pattern, and for the highland Wnt pattern, which generates 2 wing patterns instead of 1, 4 wing patches instead of 1. And the exact same thing happened in both species, generating this amazingly parallel hybrid zone with this new hybrid form in the center. So basically, this new hybrid form combines the highland variants at 2 genes with the lowland variants at 2 other genes. And this is how we get this new subspecies. And so in the center, the birds learn to avoid this new color pattern. Basically, it's just like reshuffling Lego bricks again. And I think this is a pattern that many recent studies have now found over and over again, that hybrid variation or introgressed variation is important, particularly in rapid speciation and in adaptive radiations, where many new species evolve very, very rapidly, and there is not much time for new mutations to evolve. But if through hybridization, a large genetic variation gets generated, this really helps very rapid adaptation and rapid speciation. And if you're interested in this topic, we wrote a review in Tree, which highlights many different cases of rapid speciation where old genetic variants, often derived from hybridization, have played an important role. And with that, I would like to thank my cichlid collaborators and my butterfly collaborators. And Many thanks to you for listening, and to Julia and Astrid for the invitation.